Biotransformation. Biotransformation is also one of the mechanisms used by certain microorganisms during the process of bioremediation. Biotransformation means transformation of one molecule to another form through the biological activities. So, biotransformation refers to the chemical modifications of a pollutant by biological means, typically through enzymatic activity of microorganisms such as bacteria, fungi, or algae. In the context of biosorption, biotransformation is the complementary process that may occur before, during, or after the process of biosorption, especially when living biosorbents like actively growing bacteria or fungi are involved in the process of biosorption. Biotransformation is a biologically driven chemical process in which organisms, usually microbes, convert one chemical compound into another. So when we are considering the pollutants, the toxic pollutants will get converted to less toxic or harmless molecules. In biosorption, this typically involves transforming pollutants into less toxic, less soluble or more easily immobilized forms. For example, certain bacteria can reduce toxic chromium plus 6 to less harmful chromium plus 3 or oxidize arsenite As plus 3 to arsenate As plus 5, which can then precipitate or adsorbed onto the biomass. These transformations occur through specific enzymes such as oxidoreductase and they require living cells in a metabolically active state. Unlike simple binding, biotransformation chemically alters the contaminant, offering enhanced remediation potential. Role of biotransformation in enhancing the biosorption. Biotransformation enhances biosorption in different ways. First is it can improve the removal efficiency as it converts pollutants into the forms that bind more easily to the biosorbent. It also detoxifies pollutants, so it reduces the toxicity by transforming harmful compounds into less harmful or non-toxic ones. It regenerates binding sites, as the molecules are not only being adhered or attached to a particular site, but they are getting converted to some other form. So once this transformation occurs, the original binding site is regenerated so that more pollutant molecules can get adsorbed on the same site. It also prevents the re-release of the pollutant as the pollutant is getting converted to some other form irreversibly. The microorganisms that are involved in this process are mainly bacteria, fungi and algae. They are known to perform biotransformation as a part of their survival strategies in polluted environments. Bacteria such as Pseudomonas putida, D-sulfovibrio vulgaris and Geobacter species have shown remarkable abilities to reduce heavy metals and radionucleotides. Fungi like Phanerochaete, Chrysosporium produce oxidative enzymes that can transfer complex organic pollutants including dyes and pesticides. These organisms employ intracellular and extracellular enzymatic systems to change the oxidation state, molecular structure, or solubility of the contaminants, aiding in their removal or further degradation. This table summarizes some of the microorganisms and pollutants they transform. Bacteria like Pseudomonas or Bacillus transform heavy metals, including chromium, mercury, and also they transform some organic dyes. The mechanism by which they biotransform these molecules include redox transformation, where the transfer of electrons occur to change the oxidation state of the metal. Also, it involves enzymatic degradation. Fungi, including Aspergillus phanerochaete, can transform dyes and certain hydrocarbons. The enzymes which are involved in these activity are lacase and peroxidase. Algae can biotransform nutrients and some heavy metals by the metabolic uptake of these nutrients and heavy metals. Yeast can biotransform pharmaceutical wastes and metals by bioconversion and detoxification of these pollutants. certain pollutants like chromium can get biotransformed by the mechanism of reduction. The resulting compound which get formed are more adsorbable. The organisms that can carry out this process include Pseudomonas, Bacillus, azodies which contain the azo bond and double bond N. They can be enzymatically cleaved to form aromatic amines. These aromatic amines can be further converted to the acids which participate in the central metabolic pathway of the organisms. The organisms which can carry out these processes include aspergillus and termites. Antibiotics and pesticides can be degraded by Saccharomyces or Pseudomonas and other organisms by oxidation or demethylation, that is removal of methyl group, or hydrolysis. It results in the formation of simpler, less toxic molecules. Types of biotransformation include oxidative reactions. Oxidation involves the addition of oxygen or removal of hydrogen or loss of electrons from a substrate molecule. Here the substrate we are considering is the pollutant molecule. Enzymes which are majorly involved in oxidation reactions for biotransformation include enzyme lactase, peroxidase, monooxygenase or dioxygenase. These enzymes can add one or more oxygen molecules. For example, it carries out conversion of phenolic compounds to catechols or quinones. Oxidative cleavage of aromatic rings in dies of the azodides, where many rings are attached together, can be carried out to form hydrocarbons which can get converted to alcohols, aldehydes or ketones. Ultimately, they will get converted to organic acid and can enter TCA cycle. The second mechanism is reduction reactions. Reduction involves the addition of hydrogen, removal of oxygen or gain of electrons. So this is opposite of oxidation. The enzymes which are involved in this type of mechanism are reductases, including chromate reductase, nitroreductase. The examples of these type of transformation are reduction of hexavalent chromium, that is Cr6 plus, to trivalent chromium Cr plus 3. Reduction of nitroaromatic compounds to amines, also a type of this reaction. These reactions of chromium conversion are carried out by desulfovibrio. The third mechanism of biotransformation is hydrolysis reaction. Hydrolysis means the cleavage of chemical bonds by addition of water molecule, breaking complex molecule into simpler molecules, and the molecules which are created are usually hydrophilic fragments, that means water loving fragments. The enzymes involved in these type of reactions are esterases, phosphatases, amidases and hydrolases. The representative reactions include breakdown of organophosphorus pesticides, that means pesticides which contain the organophosphorus groups, or hydrolysis of ester and amide bonds in pharmaceuticals. For example, parathione can get converted to paranitrophenol plus dimethyl phosphate via esterase activity of flavobacterium. Next mechanism of biotransformation is methylation, that is addition of one methyl group, that is CH3, or demethylation, that is removal of methyl group. The enzymes which are involved are methyltransferases and demethylases. These enzymes can carry out methylation of heavy metal like Hg2 plus mercury can get converted to CH3 Hg plus. Demethylation of methoxy substituted aromatics, that means aromatic compounds which have methoxy group OCH3. From those molecules, CH3 group can be removed. Methylation of mercury by sulfate reducing bacteria. For example, desulfovibrio desulfuricans is one of the examples of this type of biotransformation. Next mechanism is dehalogenation. Dehalogenation is the removal of halogen atom like chlorine, bromine, fluorine from the halogenated organic compounds. Enzymes which are involved are dehydrogenases, which include hydrolytic, reductive, or oxidative enzymes. Examples of these type of reactions are reductive dechlorination of chlorinated solvents like trichloroethylene. From this molecule, chlorine can be removed. Oxidative dehalogenation of pesticides and insecticides can also be carried out by these type of microorganisms. Next mechanism is aromatic ring cleavage. This transformation involves the opening of aromatic ring through oxidative enzymatic activity, a critical step in degrading stable recalcitrant organic compounds. In this type of reaction, in the first step, the aromatic rings are broken down by addition of oxygen or other small moieties. The enzymes which are involved are dioxygenases, for example, catechol 1,2 dioxygenase, which will add 2 oxygen to the ring, aromatic ring, and catechol 2,3 dioxygenase, which will add 2 oxygen atoms on second and third carbon atom each, one on each. The reactions include catechol ring cleavage via ortho or meta pathways, breakdown of benzene, toluene, and xylene. The breakdown of catechol produces cis muconic acid, which is carried out by the bacteria Pseudomonas putida. Factors that influence biotransformation include different factors such as microbial strains, pH, temperature, oxygen availability, and the presence of electron donors or acceptors, as most of these reactions are uh, redox reactions. For redox transformations, the availability of suitable redox partners, that is organic carbon, nitrate, or sulfate, is critical. Some microbes only perform certain biotransformations under anaerobic conditions, where others might require aerobic environments. So, we need to provide the suitable environment for the organism to do biotransformation effectively. The pH is also one of the crucial factors because it decides the movement of some ions or production of some ions. Also, it will decide the growth of the organisms, and organisms can grow only at definite optimum pH and temperature conditions. So, we need to consider both these factors crucial for the process of biotransformation. The genetic and enzymatic machinery of the organism also determines its ability to transform specific contaminants. We can also use some of the genetically modified organisms whose genetic makeup has been designed so that they can carry out these biotransformations successfully. Additionally, the chemical structure and concentration of the pollutant influences whether and how efficiently it can be biotransformed. Environmental applications. Biotransformation based biosorption has been widely applied in wastewater treatment, especially for the detoxification of hexavalent chromium, arsenic, and mercury. In groundwater remediation, biostimulation of native microbial communities has been used to reduce uranium and other radionuclides to less mobile forms. Biostimulation is the process where the growth of native microbial communities is enhanced by addition of certain nutrients or by controlling the processes which are required for their growth. In industrial effluent treatment, biotransformation can break down complex organic compounds, making them easier to remove by filtration or adsorption. The mechanism is also gaining attention in pharmaceutical waste management, where microbes degrade endocrine disrupting chemicals and antibiotics. These applications demonstrate biotransformation potential in both metal and organic pollutant treatment.